Hello, my name is Ross Barnett, and I will be presenting on the journal article known as Bioinformatic Expansion of Boresins Uncovers Transacting Peptide Backbone in Methyltransferases in Bacteria. So after that mouthful of a title, let's jump into some info regarding the background of the article and some basic info revolving around the various aspects of this article. You're probably asking yourself, what exactly is N-methylation? Well, N-methylation is one of the simplest chemical modifications that can occur on peptides and proteins of prokaryotes and higher eukaryotes. This peptide protein modification results in the transfer of a methyl group onto the amide group of a peptide or protein. If you move your attention over to the right side of the presentation, you will see a simplified mechanism of N-methylation. During this mechanism, the amide group is deprotonated via generic base. After this deprotonation occurs, the enzyme known as N-methyltransferase will catalyze the reaction, causing the cofactor S-adenosyl methionine, or SAM for short, to lose its methyl group by donating the methyl's alkane bond back into the electron-poor sulfur atom present on SAM. This ultimately leads to SAM becoming S-adenosyl homocysteine, or SA for short. The deprotonated amide group present on the peptide chain can then act as a nucleophile, attacking the electrophilic methyl group, thereby allowing methylation of the nitrogen atom to occur. Now that you understand the N-methylation aspect of this article, let's take a look at what boresins are. So boresins are technically peptide macrocycles that produce peptide precursors that possess the enzyme N-methyltransferase. Now, if you remember from the mechanism I showed in the previous slide, this enzyme catalyzes the overall reaction regarding N-methylation. Without this enzyme, an organism would not be able to efficiently methylate the amides present on their peptide chain backbones due to the energy barrier for this reaction being far too high. However, if the organism is a part of the boresin family, meaning they can produce the peptide precursors that possess N-methyltransferase, they would be able to efficiently methylate these amides present on their peptide chain backbones by overcoming this energy barrier. I have provided a figure to the right of my presentation, which was taken directly from my journal article. This figure is showing a boresin-type macrocycle in action with a peptide chain amide group reacting with SAM with the help of the enzyme N-methyltransferase, ultimately producing the compound known as omphalocin A. The next thing to understand with this research article is that the only certain organisms can actually utilize N-methylation and thereby be a part of the boresin family. N-methylation usually only occurs ribosomally, meaning the peptide precursor containing the enzyme N-methyltransferase is produced via the ribosome. There are three pathways that these precursors are usually synthesized from. These pathways and gene sets are denoted as class 1, class 2, and hybrid. Now, if you would take a look at the figure located on this slide, you will see that each pathway has a different set of genes. However, once again, only specific organisms possess these genes, thereby only specific organisms can utilize these pathways. Past research proved that fungi were the only members of the boresin family. However, this research article set out to prove that bacteria now can share the same title. And I have implemented a couple of pictures just for a laugh. Now that we understand the background, why was this research conducted and this article written? This research was conducted because N-methylation serves as a very advantageous reaction, especially when coupled with pharmacology. N-methylation can lead to more efficient drugs due to this phenomenon leading to improving the oral bioavailability, lipophilicity, and aqueous solubility of peptide-based drug structures. If scientists could prove that bacteria utilize this biochemical reaction, they would be able to more efficiently incorporate it into modern-day pharmaceuticals, because bacteria are much easier to work with and also much more abundant in nature than fungus that utilize this reaction. On top of this, the scientists who conducted this research wanted to prove that there are many more metabolic pathways that lead to N-methylation rather than the ones previously linked to the three classes listed in the previous slide. So how was all of this research actually conducted? A major focus of this research was the utilization of bioinformatics in order to statistically determine how common N-methyltransferase was in bacteria. In order to do this, genome mining was utilized in order to locate the specific genes linked to the boresin families. Once these genes were located, the bacteria was isolated and their N-methyltransferase enzymes were isolated via exclusion chromatography, which we talked about previously in the semester. These enzymes were then reacted with cofactors and pre-synthesized polypeptide chains with specific amino acid sequences in order to see how the N-methylation occurred and whether the bacteria in question could be accepted into the boresin family. In order to study the methylation patterns, the enzyme glucosamine and trypsin were utilized to cleave the chains to a more manageable size. The statistical data was finally recorded and then compared to past data. Overall, the research methods and materials utilized were very efficient in proving the researchers' hypothesis. Looking at some of the data provided via the gram-negative Paraglacialia arctica, a few things can be noted. The first thing to realize when viewing this figure is that the polypeptide chain was cleaved utilizing the enzyme glucosamine. The second thing to note was the organism was efficiently able to code for and produce the enzyme N-methyltransferase via the genes it possessed. The final thing to note with this figure is N-methylation occurred at the amino terminal present on the histidine and the amino terminal present on lysine. With this data provided, it could be concluded that this bacteria belonged in the Boracin family, and this data could now be recorded for future reference. The other conclusive figures provided in this article were almost all identical to this figure, but relied more heavily on the actual genes present in each type of the bacteria, rather than looking at the biochemistry itself. A few things were concluded from this study. The researchers who worked on this project were able to determine that there are over 1,500 boracins rather than the previous 100 known. The researchers have now also been able to regroup the boracin family into two classes that were then further divided into 6 distinct types. On top of this, they were able to efficiently prove that bacteria belonged in the boracin family due to their ability to produce and utilize N-methyltransferase. The final thing that was denoted was the bacterial N-methyltransferase substrate binding is much more relaxed in bacteria than it is in fungus. With all this evidence, I believe that the future of medicine is bright. Now that we better understand how N-methylation occurs and that what organisms utilize this phenomenon, plus what genes are responsible for it, we as a society will be able to develop much more efficient drugs and even lower the price of medication. Thank you very much and have a wonderful day.